Good morning, everybody. Just before Dr. Swerdlow formally introduces Professor Flint, I'd just like to say personally, welcome to UCSD. Thank you for being here. And I'm really hoping we had a great discussion yesterday about interfaces of engineering, bioengineering, and mental health issues. And that's an interest of yours also. Not what you're talking about today, I don't think, but a conversation I hope we can continue as well. So welcome, thank you for being here. So Neil.
Morning, everybody. Welcome to Grand Rounds. Dr. Jonathan Flint is professor in the Department of Psychiatry and Biobehavioral Sciences at the UCLA David Geffen School of Medicine, and senior scientist for the Center for Neurobehavioral Genetics at the UCLA Semel Institute for Neuroscience and Human Behavior. Dr. Flint completed his medical and psychiatric training at the Maudsley and Bethlehem Royal Hospitals in London and spent about 25 years as fellow and faculty at Oxford before joining the UCLA faculty about a year ago. His research uses novel approaches in rodents and humans to understand the genetic architecture of complex traits in psychiatry, focusing mostly on anxiety and depression. His CV lists about 280 peer-reviewed publications. He's a fellow of the Royal College of Psychiatrists and the Academy of Medical Sciences and was awarded the Gold Medal from the Genetic Society in the United Kingdom. Today he'll be speaking with us about the genetics of depression. Please join me in a warm welcome for our Grand Round speaker, Dr. Jonathan Flint.
Well, thank you for having me. It's a great pleasure to be here. So what I'm going to be talking to you today is not necessarily a good subject for 8 o'clock in the morning, which is depression, misery. And I'm going to start by just giving you a brief overview of why this is something that's worth studying, and then describe the strategy that we've taken to understand the causes of depression and some implications of that research. So let me just start, for those of you who are not clinically trained, to give you some idea why this is an important thing to do. And there's a wealth of epidemiological data. I'm just going to summarize these very briefly. Number one, leading cause of disability, predicted to become so by 2020. That's a World Health Organization. We had a World Health Organization Mental Health Day a week or so ago, and that reaffirmed that this is an extremely disabling condition. It's not just disabling. One person will die of suicide every 30 seconds somewhere in the world, and that's mostly because they're depressed. So not only is this disabling, it also carries a certain morbidity with it. There are 245 million prescriptions for antidepressants written here every year, costing this economy about $10 billion. And that's despite the fact that only about half of the patients that we see on those prescription medicines see much improvement. And then perhaps most concerningly, there have been no novel or more effective antidepressants developed in the past 25 years. So this is common, disabling, carries a morbidity. We don't know how to treat it. And despite all of those things, the understanding of what's going on is still extremely limited. So if we're going to change the situation, we really need to understand more about what causes depression. And I'm going to talk a little bit about that next. I'm going to start just by giving you an example from the UK Biobank. So the UK Biobank is a really unique experiment where 500,000 people have been recruited across the United Kingdom. They have been asked to give a large amount of their time, a couple of hours for initial assessment and then follow up. And you can download all of these data for free, you just need to put in an application. And all 500,000 of them have been genotyped. The first 150,000 genotypes are released. Already the next 350 are being released in the next few weeks, all freely available to any researchers. I think it costs about $150 for an application. That's about all for the processing. And clearly this is a resource for anyone looking at a common disabling condition. So it's a great place just for us to get a little feel as to how work is going on in depression. So I'll just show you one result really from this study. And what we're trying to do here is understand the genetic causes of diseases using standard genome-wide association strategies. So this is actually data from a paper by Joe Pickrell. And we're showing, for those of you who haven't seen these graphs before, this is there's a association significance on the vertical axis and along the horizontal axis of the chromosomes. And anything above this dotted line here, we think as being significantly associated. So these are results for Alzheimer's, coronary artery disease, and type 2 diabetes. And these large peaks here show that it's a successful way to find the genetic causes of these common conditions. And you can see I've left major depressive disorder blank so that you, in when I show you the next slide, can work out how successful people have been in finding the genetic components of that condition. So that's, I think, a fairly dramatic demonstration of the difficulties that have faced sorting out the genetic component of depression. So that it raises the obvious question as to why that's been so hard. And I'm going to go through what I think are the obvious explanations. The first of those is, well, it's a psychiatric disease, and everyone knows psychiatric disease is difficult to study and genetic approaches won't work. That's an argument we can easily dismiss now with the results most recently from the genome-wide association studies of schizophrenia. So this is just like the graphs I've shown you before, chromosomes on this axis, likelihood here. Anything above the red line is significant, and as it says in the title here, 108 loci that they think are involved in schizophrenia, and schizophrenia clearly is a psychiatric disease. But just because it's psychiatric doesn't mean it won't work. Second issue might be, well, it's not very heritable. Well, we know a lot about the heritability of depression. It's common. There are many twin studies. I'm showing you here a summary from my colleague Ken Kendler, who provided, this is from year 2000, a summary of what he regarded as the best designed twin studies at the time. And the vertical notch here indicates the estimate of the heritability. The horizontal line is the confidence with which that is estimated. And down here is a summary at the bottom. And the result shows that it's a heritability about 37% with a very tight, for these studies, a tight confidence interval. And to put that into perspective, the heritability for diabetes is about 40%. And as I've shown you in this first slide from the biobank, mapping diabetes has not caused a problem. So the heritability is certainly within the limits that we ought to be able to see something. So the last possibility is maybe it's not one disease. Maybe we're dealing with different conditions. And I want to give you just a couple of pieces of evidence to make you think about this possible explanation, one genetic and one environmental. So this is, again, twin study data, again from my colleague, Dr. Kendler. This axis here represents the heritability for males in red and females in blue. And you can see there's a difference in both these studies. These are very large samples. There's 3,000 twin pairs here for males and females. And this is an even bigger study, so about 30,000 altogether. The confidence intervals on these estimates are tight. You can see that it will agree with the previous estimates I showed you, that the heritability for both is about 37%. This difference is significant because these are large sample sizes. So the heritability in women is higher. But the reason I'm showing you these figures is because of the green bar here. And that represents the estimate of the genetic correlation between men and women. And if the same loci were contributing to depression in men and women, this should be 100%. And it's nowhere near that. It's more like 55, 60%. So in other words, something like 40, 45% of the genetic contribution to depression is not common between the two. So from a genetic point of view, it's hard to say this is exactly the same disease. The second way of thinking about this is from an environmental point of view. So most of us are familiar with the consequences of a stressful life event. So that, for most scientists, is sending off your life's work to a journal like Nature and having it sent back within 5 minutes. It's not even worth sending it out for review. Or even worse, getting those reviews back saying that this is the worst piece of work they've ever seen. And we all know how painful that feels. And for some people, it's hurts. Stressful life event. And we know that the sort of low mood that can be a consequence of that. And if the stressful life event is severe, we're all familiar with the fact that certain people would become depressed. But what's important to know here is that the relationship between the stressful life life event and the episode of depression has a fairly well-defined temporal relationship. That is to say that if, and I'll put a big if around here, that if this is a causal relationship, then this is an interval of about maybe 2 or 3 months. So if you come to see me, I'm your psychiatrist, and you tell me you're extremely depressed, and I go through stressful life events, and you say, last year I had my grant rejected and my papers rejected, that's not likely to be the cause. If it's something that happened in the previous 3 months, then I'm more likely to take it seriously. However, there are other forms of stressful life event which do not show that temporal relationship. And the most important of those, and most well documented, is childhood abuse, particularly childhood sexual abuse, which increases the risk of depression more than anything we know. 10, 20-fold increase in the risk of depression. And the important point about this, in terms of what I'm arguing now, is that risk is spread across the lifetime. So it's not that you'll find the same temporal relationship you do in adult life. So that would indicate that whatever pathway is operating between these two events is not the same pathway that's operating here. So it makes it hard to consider this really as being the same causal factor. Something else is going on here. So those are just 2 bits of evidence to make you think a little bit about whether we're dealing with a single condition or not. And there are implications clearly for that when we carry out a genetic mapping experiment. So given that, I spent some time talking about how we might do this with my colleague, Dr. Kendler, and we decided that we should take this sort of information seriously and try and identify all of the features that would make us work with a homogeneous sample set. And those things that we couldn't exclude, then we should at least measure so that we could include in our analysis and take into account those effects. So the design we came up with is as follows. We chose 6,000 cases and 6,000 controls, and we didn't choose those for particularly good reasons. I'll tell you why we chose that number. So this study was designed before the first genome-wide association study came out. And that, if you recall, was about 2007. And the person leading that study at the time was Peter Donnelly. And I met Peter before the results had came out. And I said, have you found anything? He said, yes, they found stuff. And I said, what was your sample size? And he said, we had 3,000 cases, and it was a larger set of controls. And I said, did you find anything for the psychiatric disease? They had bipolar disorder in the group. And he said, no, we didn't. So I thought, well, okay, we'll just double. I mean, really, that's all it was. We didn't have anything more to go by. We chose to work with women only, and for the obvious reason that you've seen that it's more heritable in women. And we wanted to exclude sources of heterogeneity. So all cases had to have multiple episodes because there's some evidence to indicate the genetic effects on a single episode and multiple episodes are different. And then all those things that we couldn't exclude, we would measure. So all of the known risk factors, personality, childhood sexual abuse, stressful life events, poor parenting, low social support, and a host of comorbid disorders, mostly anxiety, that we could assess. And in order to do this, we decided we needed to work with a very large population. Because although this is a common disorder, by the time that we've restricted it to one sex, recurrent depression, certain age groups, it's beginning to cut down the numbers. And we needed a population which had a really good health system where we could work with the doctors. And as you can see, we ended up deciding to work in China. And the way this was set up is that myself and Ken would go to China. And I'm no expert, at least when I started this, I certainly wasn't, in how to assess psychiatric disorders for research. This is what Ken has done all his life. So we go to China. Any of you who know Ken will realize that's quite a statement. No, China's been many challenges we had to face, I have to admit. And working there has not been easy. But the training of doctors turned out to be something that I think we were both very proud how well we did with this. We provide everyone with a laptop, and what everyone's holding here is the Bible for this study. This is the description of what questions you should ask. And our aim was to make sure they didn't deviate from the questioning. And we would have a series of practice interviews, either between the interviewers themselves or bring in practice patients, which we then assess. And about 80% of the subsequent interviews with patients are recorded, uploaded to a server, and then a group of the doctors that we've been working with for a time, whose opinions we trusted, would then listen to a proportion of these interviews, report back on the quality, and edit if appropriate. So we began this in 2008, and I just want to show you this slide, which gives you some indication of the problems that we were facing. So the black line here is the cases, and the red line is the controls. And the first thing to note is that there's a sort of different slopes as you go along here. And if you notice, when we started, we were in Shanghai with about 8 hospitals. And by sort of a few months into the study, you can see that the slope of this is really not looking good. And we were projecting taking maybe 10 years to finish the study. So because we're in China, big advantage is it's such a big country that really all you need to do is go out and speak to more hospitals, get them interested in your work. And then when you've done that, the slope of the graph improves for a little bit, and then you can see it sort of drops off a bit. So we have to recruit a few more hospitals, and it carries on for a while. So that's how we maintained the rate, just continuing to go out and discuss with hospitals, involve them in the study. The other thing you'll note are these horizontal lines here, which occur at regular intervals. And when those first happened, I have to say my heart sank. Why on earth was nothing happening for 2-week intervals? And of course, the answer, those of you who know anything about what happens in China, is everyone goes on holiday. So this is the Chinese New Year period, when basically the entire country shuts down. And then we managed to finish really on time using that approach. Now, that did require for us to recruit a lot of hospitals. So this is a sort of picture of where we had to, hospitals we had to speak to in China. There were, in fact, about 30 cities whose hospitals contributed, and we had samples in the end from 59 of the hospitals spread across China. And in order to do this, as I pointed out, I did have to go to China quite a lot. I did have to speak to many of the hospitals and more hospitals, and even more and more and more hospitals and more hospitals. And it's a quite tiring exercise, as you can see. So that's 5 years of my life in 6 slides. When we'd finished, we decided that we would get the genotyping done in China. So we came to this place in the far south. This is in Shenzhen. BGI stands for Beijing Genomics Institute, or used to, but actually it's 3,000 miles away from Beijing. And it's still, I think, certainly was then the world's largest sequencing center. When I came to them and said, we've collected this large sample, would you be interested in helping us with the genetics? They said yes, and they said, well, why don't we sequence everybody? Which I thought was a great idea. No one had done anything like that, certainly on the scale we had. But of course, we didn't have really quite enough money to do the sequencing. So we ended up with a rather unusual strategy, which I've summarized in this slide here. So this is the sequencing depth. So those of you not familiar with DNA sequencing, the way it works is you chop up the DNA into small fragments randomly, and then you just, as it were, pick them up from the floor at random and sequence them. And in so doing, of course, you don't know where any particular part comes from. So to cover the genome, you just have to do it many, many times. And typical genome sequencing studies would have a 30x coverage. That will be somewhere down here. And our mean coverage is at 1.7x. So it's a very low sequencing coverage. But our argument was that this should work because we were really only interested in catching common variation, which we made the assumption this was going to be the major contributor. And the other reason I thought this was important to do is because although this is the world's largest ethnic group, at the time, there were only about data from about 100 people at high depth to give us what the common variants were. And us doing this, of course, gave us everything. We had 20 million variants from so doing. But our problem with the data was that we would generate from each person something that looked a bit like this. So these are the 2 chromosomes. In many cases, we have no data at all. Sometimes we get both chromosomes. Often we only get one allele. So we have a missing data problem. And what we need to do is turn that into something that looks like this. And that's a computational problem because of the correlational structure of the human genome. If you have a C at one position, you can predict with a certain accuracy that you'll have a G at another if you have the haplotype structure of the population to hand. And we were able to generate that and indeed to turn these data into that, except, of course, rather than it taking a couple of months, this process took about 2 years because we had 30 terabytes of data and quite a lot of QC to carry out, which I'm not going to go through. So having generated our genotypes, we then carried out the genome-wide association, and you should be able to interpret the results from what I showed you before. Likelihood here, chromosomes, this is the significance threshold, and there are 2 loci which exceeded the threshold. Now, anyone in this field knows now that you're not going to get this published in a high-quality journal unless you can show that you can replicate the results. So I wasn't about to go and spend another 5 years collecting a sample, but fortunately there was a group I discovered in Beijing who had independently been collecting, not as large sample and certainly not as well defined as ours, but they kindly let us have a small quantity of their DNA to try and replicate our findings, and I'll show you those results next. So this is a summary of the data from our initial study. These are both of the loci on chromosome 10, and they're both relatively common variants, one with a frequency of 45%, one at 26%, and these are the p-values that we obtained. And the data that we got from my colleagues was a smaller sample set, over half the size. Both loci here was significant, and the combined ratios over here, so now we're up to a sample size of 17,000. So that I felt, as much as these things go, was a good enough replication. But one thing I haven't mentioned to you, and we'll talk about this a little bit, I'll talk about this in a bit more detail in a moment, was that there is a worldwide effort, the Psychiatric Genomics Consortium, who are similarly working on this problem. They had a sample set of 17,000 and haven't found anything. So, of course, an obvious thing to do is ask, well, what are these loci doing in the Psychiatric Genomics Consortium? And this is a comparison between the two. So these are our data here with the p-values, and you can see the p-values in the PGC data set are nowhere near significant. But you will also notice something else here if you look carefully, which is these are the IDs of the loci that we mapped, and we don't actually have the same loci in here. And the reason for that is because they don't have data for these loci, because it turns out that both the loci that we were interested in are very low frequency, particularly this one. It's a frequency of 45% so it's really not really comparable. And the power you'd need to detect this, of course, means that you'd need sample sizes well in excess of what they've got here. So I'm not really sure how useful that comparison is. But there's some other issues around that we'll deal with in a moment. Now, there's another thing that happened recently, which is that those of you who've read the literature will know that end of last year, there was a paper published in Nature Genetics with this title, saying that they'd found 15 loci. Again, this is not PGC data. This is from a company, 23andMe. This is what their plot looks like. The chromosomes again, and likelihood, and you can see there's not actually 15 because this is their discovery sample, and they up the numbers. But there's not a figure showing all, so I'm just showing you this figure. But you can see there's some really significant things here. So then an obvious thing to do is say, okay, well, let's just see what that looks like in comparison with the data that we've got from our CONVERGE study. So I plotted out here in red, these are the 23andMe loci where I could find data, and this is the results from CONVERGE. So you might think, well, maybe it's just a question that this really doesn't look very significant, but maybe it's a question of them being perhaps they're in the same direction. We can support these results by seeing whether the allelic effect's the same. That turns out not to be true, as I'll show you in the next figure. So here I'm showing you the comparison at each of these loci for CONVERGE and for the 23andMe data. This is in terms of the odds ratio. So if forget the significance of this, if the loci have the same effect, they're going in the same direction, then they should both be the same side of 1. So 1 is no effect at all. So what we should be seeing is things where both are the same side of 1 or other side. So this would be a case where they're both operating in the same direction. As you cast your eye across this, very many of them, that's another one, look like this, one going one side, one the other, one going one side, one the other. It's random. There really isn't a pattern here. So none of these loci either have a significant effect or go in the same direction. It doesn't match up. So why is this? Well, let's just think again about what I was discussing with you earlier about what we mean about a single disease. So the first thing to consider is what is major depression in this sample. And the diagnosis that they used, there's a number of them because the data come in different cohorts, but they're basically all a self-report answer to a single question. So I spent some time at the beginning of my talk explaining to you that we went to China, we taught doctors for a week, we gave them a booklet, a laptop, they had a 2-hour interview, we then spend a lot of time editing that to make sure that we were diagnosing depression. Whereas this group sent out a question, are you depressed? Now, clearly that has some information, but we really don't know how that maps onto the sorts of things that I've been considering as depression in China. And the genetic data I've just shown you, in terms of the overlap, at least the significant effects, would indicate it's not the same. But let's pursue that question a little further. Maybe there's some other way that we can get at this. And let's start off with just, this is the correlation in the heritability between, so I've shown you that UK Biobank didn't find anything, 23andMe did, this is us, this is the PGC. This axis here, the diagonal, is the heritability for each of these studies. And as you can see, CONVERGE, not as high as the heritability that we estimated from twin studies, but that's fairly typical of these approaches. This is based on the genetic data alone. But the interesting part of this graph is to look at these genetic correlations. So PGC and UK Biobank seem to be mapping the same thing. The correlation here is 0.96. CONVERGE and 23andMe, the correlation is, well, almost ignore it. And our correlation with most of these other things similarly is very low. So there's, again, I think from a genetics perspective, not enough evidence to say that we're really dealing with much of an overlap here. So I want, for the last part of the talk, just to deal a little bit more detail with this question of whether we're dealing with one disease or not. And I'm going to take one example and push that a little bit. And the example is based on the environment again. Remember, I talked a bit about this to you before. So these are the stressful life events that we ask in our study. It's a fairly standard list of events. And if we ask what effect they have in CONVERGE, the answers are given here. So this is the association p-value with the risk of depression. But importantly, look at this column here, which is the odds ratio. And the first point to note is that these are consistent with what has been reported elsewhere in the world's literature. The second thing to note is things that you would regard as being more serious, the obvious example here is rape, has a much higher odds ratio, consistent with it having a causal role in causing depression. So these data are reassuring. It's what we'd expect to see. Other people have seen them. So we're probably getting the right thing. Now, the next thing to ask is, what would you expect if I take all of the people in our study who report that they've suffered life adversity, and I just take them separately, and I map the loci that contribute to that form of depression, compared to people who don't report any life adversity. There's no indication. I got depressed, doctor, but I really don't know why. It just came out of the blue. I don't understand. It's been happening many times, and there's never a precipitating event. So if I take that group of people and compare them to those who've had a stressful life event, what do you think we should be seeing? So we're going to take out the subjects who experience stress, and we're going to rerun the genetic analysis. And this is the result for subjects where we remove subjects who've experienced stress. And what you can see here is we've reduced the sample size. It was over 10,000. So we've chucked out almost a third of the sample size. And we find two loci here, not apparent in the original study. Again, one here. And although there's some signal from one of the loci we had on chromosome 10, the other one has almost disappeared. And just to put that in perspective, here's what we saw in the entire sample set, and here's what we see in the reduced sample set. So why would that be? And our argument, of course, is that actually what we're tapping into is something with a different architecture, that people who don't report a stressful life event will have an increased heritability, and the genetic effects will be different from those who do. So if that's true, then there's a number of implications. I just wanted to, the next slide is just dealing with an issue as to whether we might see these effects by chance. So if you just randomly selected 7,000 people from this group and run the analysis, what would we see? So we ran this across the loci. This reflects what we see in the real data, and this reflects what you'd see if you randomly selected 7,000 people. And clearly not going to happen by chance. We can assign a p-value to that from the empirical distribution. The other thing, as I mentioned, is that we'd expect a change in the heritability in those two groups, and that's exactly what we see. So this is our results from the full cohort here, and I would like to draw your attention to this component of the graph here, where we have the heritability estimates. Now, we estimated it in different ways, and that's because there's lots of discussion about how you should do this adequately, whether you should take into account the linkage disequilibrium structure, and I'm not going to go into the details, only to point out that these estimates are pretty consistent here. This is for the full cohort. And just draw your attention to the adversity exposed and non-exposed groups. And you can see that this is indeed, actually, both of these are higher than we get from the exception of the LDEC estimate. They're higher than we're getting in the full cohort. So an explanation for this would be that we'll get some dilution effect because the loci are being spread about across the entire cohort. So it's consistent. It doesn't prove it. These are the estimates are broad here, but it's consistent with this having a heterogeneous genetic background in the full cohort. So it's a hint that we are indeed looking at something with different genetic architectures amongst those who do and don't report stress. And to test that specifically, we looked at the loci here. These are the 4 loci that we find in our stress and unstressed groups. These are the 2 loci in the full cohort. And we run a test here for the heterogeneity of the effect. So this is taking the 2 groups and combining them in a meta-analysis, but allowing that locus to take any effect it likes, whether it's going up or down, and testing for that effect. And the important point here is that 3 of these loci are showing a high degree of heterogeneity, highly significant degree of heterogeneity between those 2 groups. So that would show at least at those 3 loci that indeed we can confirm that the effects are not in the same direction. So this was all sort of intriguing to us, and it did indeed suggest that our thinking was on the right line. And so an obvious thing is to go back to the UK Biobank and say, well, maybe they screwed it up. Maybe the reason that they're not seeing anything is because they didn't take the heterogeneity into account. Now, the problem with the UK Biobank is, although it's, they have a lot of data, they don't have a lot of information just on depression. But we were able to take the data from major depressive disorder, and we could subdivide it in a couple of ways. And I want to show you one of those now. And that is by dividing it by sex. So remember I said that we think the genetic effects are different between men and women. We think the heritability is higher in women. So as you can see here, what we've done is simply divide the UK Biobank into males and females. And what we found was that. This is again consistent with the idea that there is heterogeneity in the sample. Of course, we can't do the same analysis in CONVERGE because it's all females. So it's hard for us to replicate this in our own sample, but at least we can replicate the finding that there's heterogeneous genetic effects. So what I've told you so far is that you can find genetic effects in this condition if you think very carefully about what the phenotype consists of and take the homogeneity into account. What I haven't told you is anything about what those loci might tell us about the underlying biology. And for the last 10 minutes, I want to just deal with that question. So here are the summary of 3 loci that we found, zooming in across the genome. And anyone who's worked in this area is familiar with the fact that you get a peak with some association signal, and typically it lies in a region here where there isn't a gene. The argument is it's probably lying in a regulatory region, which could be in an intron, as this one does, or it might be in an enhancer, some active region which operates at a distance from its cognate gene. And this makes it very hard to use the actual position of the locus as a way of saying exactly what that gene is. But it's clearly at some level it's correlated. So I'm putting that in a proviso before I tell you. The one thing we noted, if we look at the closest gene, then there was a surprising degree of similarity in what they were pointing to. So these are the three closest genes of those loci. And sirtuin is an NAD plus acetylase, which is involved in mitochondrial biogenesis. LPGAT1 is an enzyme localized to the inner mitochondrial membrane, and this transporter is mitoferrin involved in the transport of iron into mitochondria. And all of that, of course, seemed completely at odds with what we'd expected. These are not genes involved in, obviously, in neuronal function. But we had one other observation that made us think hard about whether that might indeed be telling us something important. When we sequence our subjects, we sequence all of the DNA. The mitochondrion has its own genome, so we sequence the mitochondria of all of the subjects in our study. And what we found when we did so was the following. I'm plotting out here the amount of mitochondrial DNA. So this is, because it's next generation sequencing, we get lots of reads of the genome, cut them up, and the number that we get is correlated with the total amount of DNA. And we can correct for the amount of nuclear DNA and arrive at a relative measure of the amount of mitochondrial DNA. And you can see that this is a highly significant difference. The cases here have more mitochondrial DNA than controls, though quite small, but because we have such a large number of people, it's a highly significant difference. And we wondered a lot whether this must be some form of artifact. We wondered whether it might be something to do with the drugs people were taking, whether it was a change in the cellular composition or some other compound we hadn't taken into account. What I didn't tell you is that DNA in our subjects was taken from their saliva. This is not blood DNA. So there could be a number of things contributing. We could investigate this because we did have drug records. A number of patients had reported that they'd taken no drugs at all. We saw no difference in this finding between those who did and didn't. And we were able to test this in mice by giving fluoxetine and a couple of other antidepressants and see whether the mitochondrial DNA from the saliva, I should say in passing that getting DNA from saliva in mice wasn't that straightforward, but we did manage it, and check whether we see any change between those who do and don't receive the drug, and we were not able to see any difference at all. A more difficult thing to rule out was the possibility that there might be some change in the cellular composition in those who had depression, and was that showing the effect? So we didn't at that stage really know what the cellular composition of the saliva was. We took some saliva from subjects and had a look, and it turns out it's mostly actually white cells. There's about 70% are white cells. And we did this analysis where we took the, this is looking at the mitochondrial DNA content of components of white cells. And this is the log normalized ratio. And you can see that NK cells here have 2 orders of magnitude less than the CD3. So it would not take a big change in the cellular composition to increase the amount of mitochondrial DNA that we were observing. So this is we thought a reasonable objection, an explanation for what we're observing. So in order to deal with that, we thought we have to get a measure of the cellular composition. We can't go back to the samples we've taken, we've extracted the DNA. So as a substitute to that, we decided to look at the methylation profile and ask whether there was a difference in the methylation profile on the assumption that the methylation profile represents the tissue composition of the sample we're looking at. So we took the extremes of our distribution. We looked at those people who have got very high and very low mitochondrial DNA, and we ran a methylation study, and I'm just showing you the results for a number of methylation marks. This is our cases, these are our controls. These are markers for blood cell types, and this is for buccal epithelial. And you can notice there's maybe a little bit more blue in this group than the others, not much. And the way we did this was then just to analyze and see if there's a difference there. And I take into account the amount of residuals of this. And the residuals of the methylation analysis are really no different than the mitochondrial MD effect. So we cannot explain this difference by the methylation change, indicating that, at least from that analysis, we don't think that cellular composition is to blame. And the last thing we could do is obviously test it in another sample and just see whether there's a replicable sample. So ruling out possible other compounds, we contacted a group in London and found that this is from blood DNA, and were able to show again in their sample there was a difference between the cases and controls. So a lot of work which I've summarized just to try and convince ourselves this finding was not due to some artifact. So if that was the case, then what might be explaining this, and how might it relate to those genetic findings that I pointed out to you earlier? And the first thing we did was just map the amount of mitochondrial DNA. We thought that maybe that might indicate whether we were seeing the same loci contributing to both. And we found two loci. It's one on chromosome 10. That's where we found two loci for depression. But in fact, this is not at the same position. This turned out, in fact, to be over the locus for TFAM. So TFAM is the transcription factor activator for mitochondria. It's the molecule that's involved in mitochondrial DNA replication. Indication the mapping is probably telling us something useful, and also telling us that whatever is genetically determining these effects is not contributing to the risk for depression. So these are probably different processes. The second locus is in an intron of a cyclin-dependent kinase here. Again, no indication that it's involved in risk for depression. So we think that's an unlikely explanation for the relationship. But what we did notice is, going back to our consideration of stressful life events, is that the people who reported more stressful life events here did have an increased amount of mitochondrial DNA compared to those who didn't report stressful life events. So that led us to think that maybe this was part of a stress response. And the way that we addressed this was to think what would happen to the mitochondrial DNA in a model organism, in mice, if we stressed them. So the experiment involved chronic stress. We had to stress the mice over about a week's protocol. One day it gets dropped into a bucket of water. Second day it gets stuck into a small tube so it can't move. Third day it doesn't get much sleep. And then it gets a small electric shock. So it's a constant stress. And then on the weekend, the student doing this gets a break before he carries on in the next week. And we did this for 4 weeks. Here are the results. So the stress mice are in red. And you can see over that 4-week period, the amount of mitochondrial DNA did indeed increase compared to those with stress and with no stress. And then there's a box here marked 8. And that's for we stopped the experiment, stopped the stress after 4 weeks, and you can see the numbers go down. So that tells us it's dynamic. So it seems to go up in response to stress, and it'll go down if you don't with stress. So it's not this cannot be a trait marker. It has to be a state marker. And we also checked in multiple tissues. These are the non-stressed. These are the stressed animals. There is a decrease. It looks like a little bit of a decrease in muscle and ovary, a large increase in liver. And we couldn't really convince ourselves that there was much going on in brain. but it was a large piece of hippocampus, so we haven't really done this in a systematic approach. So there's clearly things going on in other tissues, but I wouldn't push that result too far at the moment. And the other thing we looked at was the effect on mitochondrial function. This is a measure of the oxygen concentration for the stressed and non-stressed. And basically what we're doing, if you think of it this way, we're adding in some substrate, some ADP. What the mitochondria are doing is turning the ADP into ATP, and when they do so, they consume oxygen, so the oxygen concentration should go down. And you can see this drops as it should do, as a marker of the efficiency of the mitochondria, but the stressed animals really don't show very much decrement at all. The addition of these other chemicals, the cyanide, are controlled. So I think this is the easiest thing to understand. We should see an increase in the slope, and we don't. So whatever is being produced by stress in terms of the mitochondrial function is less efficient in terms of oxygen metabolism. So I'm going to leave it at that point because we still don't really understand those results, but I'm going to point out to you that we've found 2 replicated loci. We've shown that the genetic analysis can identify sources of phenotypic heterogeneity, which I argue underlies both the successes and failures in this field, and that the amount of mitochondrial DNA is increased in response to stress. The full relationship of that and to the genetic findings is something we're still trying to sort out. And I'll finish just by thanking all of the hospitals in China that have contributed to this, and my colleagues in Oxford and Ken Kendler in Richmond. I'll take questions now. Thank you very much.
Well, thank you very much. Wonderful presentation of a rather complex topic. So, are you open for some questions then?
Of course I am.
All right. So, we have some people who are always first. Professor Barbara Perry.
Well, I commend you for your systematic efforts in obtaining this enormous amount of data. In the other male data available, I'm wondering, can you tease out whether there's this effect of stress-responsive depression and non-stress responsive depression?
Well, of course, that's what we'd really like to do. The difficulty is that no one has got the data that we've collected. So the psychiatric community has gone over to the view that you don't need to phenotype anymore. You can just go and chat with somebody, and they've got whatever condition you want, and not take it any further than that. So the number of people who've got the data that we need is very limited, and they certainly don't have it in the sample sizes that we need. So these are not big effects we're seeing here. I mean, we're seeing something in a sample size of 6,000, which I'm very surprised we saw, to be honest. This is not what's being reported in other psychiatric conditions. But there is no one in the world at the moment who've got this type of detailed information that we need to really push this further. So not at the moment. If you have them, I'd be very keen to see. I mean, anyone.
I'd be curious at some point to hear about the problems you had working in China.
Oh, okay.
The rest of us.
How many weeks do you have?
So, Jonathan.
I'm curious, in your mitochondrial analysis, first of all, do the cases and controls differ systematically on body mass index? And did you find a correlation of body mass index?
There is a small correlation, but it's not significant. We couldn't explain it by that. We have like 1,000 different variables in our data set. The only thing that showed any correlation were these stress measures. Because the odd thing about them is this study was not set up. This was a genetic study, so we're interested and asked about lifetime history and your worst episode. What we didn't have was a measure of current mood. Why would we? We're looking at genetic effects. And everything that we got from the mitochondrial DNA suggested that this was probably a reflection of current state. Because I've shown you from the mouse data, it's dynamic. So there were probably people who were reporting a very severe history of depression that didn't have raised mitochondrial levels because they were at that stage of being well for maybe 6 months or a year.
Independent of things that would differ between cases and controls, just in general, are there variables that do correlate with mitochondrial levels?
Well,
but maybe the same in cases and controls, but still correlate.
I mean, well, you see this in controls just as much as you do in cases. It's not that it's not there. I mean, anyone who's what we're seeing is something that seems to be response to stress. So if I give you all and the other thing, it's got to be chronic stress. It's not just like a one-off event. And that was the other thing that we saw in our data. It was applying it, and the mouse experiment made that pretty clear to us.
It seems that I would expect that age might correlate.
Not much. It made a little bit of a difference, but not much. But I mean, we've got a pretty. We have people between the ages of 40 and 60. We'd specifically chosen and they were pretty tightly. So the median was 47. Where there may be effects there, we just don't see them.
I just might comment that in the HIV studies, neuro-HIV studies, also these mitochondrial DNA levels correlate with inflammatory markers, even in people who are well controlled, and some relationship to cognitive impairment. That's just an aside. Sorry.
Fantastic talk. I'm 10, 15 years of remarkable activity. One of the inclusion criteria that you mentioned was that these individuals had to have multiple episodes of depression.
Yes.
So sort of a multiple part question. One is that a common inclusion criteria amongst the other studies that you mentioned?
No.
And is there some thought about whether mixed in there are individuals with cyclic mood disorders, or were there individuals who had like a high number of recurrent episodes that you could distinguish from those who had just two? So yeah.
So no, people have not done this because for a couple of reasons. One reason is that if you restrict it to that number, you cut down the numbers, so recruitment drops. Another reason why we think China is a good place to do this, because you do need a large sample size to get it. And the other reason is because single episode depression is commoner than recurrent overall. So there's an argument to say, well, we want to get the common disease, so we should do that. We took the view that from this argument about it being heterogeneous, we should concentrate on recurrent depression, and from some evidence indicated that that was more genetically determined. The question as to whether we would see a heterogeneity amongst those who, let's say, have 2 or 3 episodes as against a larger number, we haven't really been able to see that, but it's a little difficult at the moment because the distribution is pretty skewed, and it's not like, so the adversity ones is relatively easy. We could just make 2 groups. Sorting out in terms of the numbers wasn't that difficult. And your other comment about cyclicity, I suspect, is pointing to whether we had, say, bipolar or other conditions in there. So we really tried our best to exclude that, of course. So we would look in the hospital records, ask the doctors to check on this, and we're getting, one of the reasons why we chose an older age group is because we wanted people who, if they were going to have a manic episode, we hoped would have developed it by the time we're seeing them. So minimum age is, median age is 47. So that seemed to us unlikely. That said, of course, what we're doing is speaking to the doctors all the time and asking them, look, are there any people that you've seen that you recruited into our study who've subsequently come back with an episode of mania? And there was anecdotally about 2 or 3% were coming back in, but this is anecdotal. I mean, we can't exclude it. I mean, exactly, we can't exclude that. But our sample at the moment is not really big enough. And there were some people looking through. So if you just take the distribution of recurrence, there are some people who reported having 96 episodes. Who knows what's going on there?
Yeah, Jonathan, I enjoyed your talk. You had a slide where you had the genetic correlation with Converge and 23andMe and UK Biobank and et cetera. And obviously Converge didn't correlate that highly with the others, which could be because of the phenotype that as you've defined it. But what would happen if we had the same kind of data about some banal thing like type 2 diabetes, where one cohort was Chinese and the others were all European?
Okay,
would you see the same thing or not?
So that's the sort of thing that Alkes Price's group and others love to deal with. And we've used some software which supposedly takes that into account. Whether it does or not, I'm not expert enough in that to know how adequate that was.
But the software, if you gave it a disease like type 2 diabetes, where presumably everyone
Yes,
phenotype it
so
the same
that
way,
has
it
been
would
done.
work
So
just
Andrew
fine.
Morris has worked on this, and I
Yeah.
talked about this with Andrew, because there's a nice trans-ethnic study they did 2014, I think now, where they attempted this.
Okay.
I think the bottom line on this is we've done as best as we could, but we can't rule out that a proportion of the difference could be due to that.
Yeah, okay. Also about the mitochondrial stuff. So you have the stressed animals, and it's wonderful that after 4 weeks of chronic mild stress, you get the change in mitochondria. And then you also had animals that were treated with antidepressant, and that didn't seem to do anything. But
No.
what about stressed animals treated with antidepressant?
We didn't do that.
You didn't do it, yeah. Because you might like to do that, right? Because antidepressants would reverse some of the behavioral effects of the chronic stress.
Yes, you did think so. Yes.
Yeah, I mean,
Yeah.
I think
Yeah,
they do.
so that would be
yeah.
worth
Yeah.
would be worth doing.
Okay.
You're right. Yes,
And then
haven't done
finally, Jerome had a paper, I think in Biological Psychiatry, about the mitochondrial stuff. I think maybe in a European group.
that. Oh
Where-
yes.
Did,
You
right?
read that paper? Oh,
I'm afraid I did. Yes, sorry.
No, not at all. No, I
It was
thought it was a
mercifully
nice
short,
paper.
but
Yeah,
but-
we just had that as a short report.
yeah. But you didn't talk about it
No,
now.
no, I can very briefly
Yeah,
describe.
you're right, exactly.
So what we did was really going back to something that Jonathan was asking about. What we did was we found a group in Germany who had collected a cohort of 400 people with depression, and this is a treatment study, so they were taking blood samples every week, and they assessed them with a Hamilton rating scale. It's a doctor's measure of how depressed you are. And so typically people with a score of 17 get treated. Anyone over that score was included in the study. And then they assess them every week, and when they get down to a when their Hamilton score drops to a certain level, then they stop treating. That's the basic design. So we thought that the mitochondrial DNA should be high when these people get assessed, because they're coming in with this mood change, and then we should see it gradually decay. That's not what we saw. What we saw was that when they were seen at admission, so that was at their worst mental state examination, so when they have the worst assessment of depression, the mitochondrial DNA was about the same as the controls. But over time, as they were followed up, the mitochondrial DNA levels increased. So my guess is that what we're seeing in the mitochondrial DNA is a sort of delayed biomarker. And remember, I showed you in the Chinese data, it took about 2 weeks to see any effect at all, and after 4 weeks, it was showing an effect. Let's say that a really bad thing happens to you, someone close to you dies, it's very likely you're going to get depressed, and that may well happen within less than 2 weeks. In fact, very often that's the case. So what we suspect that the mitochondrial DNA represents is more a measure of the biological consequences of you being depressed. And it has the interesting implication that maybe, given that we're treating people with a biological therapy, we shouldn't be targeting what they're telling us about their mood, but maybe we should be targeting what we know about the biology of their condition. In other words, could we treat them on the basis of the amount of mitochondrial DNA as a surrogate rather than what people are saying? Which of course would mean that when the patient comes in and says, I'm perfectly all right, doctor, and we check the mitochondrial DNA and we say, no, you're not, keep going with your tablets, it's going to cause a bit of argument.
Thank you for giving us such a comprehensive, very labor-intensive, hard work. We work in China too, and but we are not on depression. We're mostly on schizophrenia and schizotype of personality. And in fact, the doctor in Changsha, I don't know whether you work there.
We work in Changsha.
Yeah, Changsha, and the graduate student from Changsha working with us now. Essentially, reliability to work in a different culture with a different language. We tested 2 sites. One is Changsha, one is Hankou. And we let them have the psychological test. And Hankou, north of Changsha, show the IQ is almost the average of 130 to 150. So we immediately exclude that site. And we zero in on Changsha. And Changsha, long-standing collaboration. So I know how intensive, how labor intensive your work. Congratulations.
Thank you.
It's I know Kendra well, and it's very typical of Kendra.
Yes.
This area of research.
Yes.
And genetic epidemiological research is always very, very intensive and collecting the big data. Now, yield is not necessarily very big, but as you show us today, at least you have some hint from this very heterogeneous group. There is a hint there. Now that is the my congratulations to you. Now is my question to you. Should I stop? From our experience studying stress, we taking the advantage of Marine going to Afghanistan. We took the genetic material before going to Afghanistan, and coming back comparing those with PTSD and those without. And those with PTSD, before going there, we analyzed the RNA. We found that the RNA profile are very interesting. It is the inflammatory gene. It is the immunological gene are compromised. So we are now looking into the potential childhood abuse of this group and any others. And another way of looking at is that those who exposed but didn't develop a PTSD, that is another side of the coin of studying the resilience. What are the resilience genes involved? This is all from genetic point of view. But of course, environmental factor are very important. So I would like you to comment on the future research on the inflammation and the immunological component of the, particularly in heterogeneous group in psychiatric condition. In your case, in depression, are you contemplating of using the immunological aspect of the underlying potential for the future treatment of some of the depression, immunological immunotherapy of depression? This is just the take opportunity of your gigantic experience to look for the future research in this area.
So sadly, we cannot do very much because all we had was a small amount of saliva and we extracted the DNA from that. So we don't really have any information about the immunological component. We don't see any of the loci that we identified being involved in immune function. So at the moment, I can neither support your hypothesis nor reject it.
Thank you.
Thanks for a really nice talk. We appreciate the way you systematically work through thinking through all the issues. My question comes back to actually kind of the beginning of your, where you started out with, saying, well, why do we have so much trouble finding genes for depression? It's not because it's a psychiatric disorder. We found it in others like schizophrenia. But it strikes me that a lot of the issues that thinking about and what in depression that might make it difficult to find the genes apply to schizophrenia as well. So I think, although the GWAS meta-analyses done on schizophrenia, it's not like everybody did nice, really careful, structured diagnostic interviews like you and Ken did, and so a lot of those diagnoses are very mixed, very sloppy, and so it seems like a lot of these same issues apply. So
so I
thoughts
think
of why it's still
it's a reasonable argument. I think there are a couple of answers, well, a couple of things to think about here. The first is that the diagnosis of a really severe psychiatric illness, particularly with people in hospital, is very different from what we're dealing with a condition which has a 40% environmental component. And the other thing to bear in mind is that even if we make the diagnosis in depression accurately, in a unselected group. We're essentially going to combine people who've had a single episode, maybe their cat died, they were feeling a little unhappy for a couple of weeks, the doctor says they got depressed, and that'll go as a case along with somebody who's had 20 episodes, ECT, and tried to commit suicide 10 times. And whether that's the same as, well, when I made my diagnosis of schizophrenia, I didn't really rule out A, B, and C as against someone who spent 10 hours, I'm not sure, because I don't think actually we're dealing with the same degree of heterogeneity in schizophrenia, but it's a reasonable question. And the other way to think about this is that probably schizophrenia, because it's been relatively easy to solve with just increasing sample sizes, maybe it does represent a more homogeneous condition, but that's just an assertion at the moment. I don't have any proof for that. So getting into, we were lucky in a way, because we didn't really know this at the time, but getting into a hospital in China is much, much harder than getting into a hospital almost anywhere else. There's a huge stigma about talking about mental illness anyway. Families will tend to look after you. So if you get into the hospital, you're much more likely to be more severely ill. Our rates of melancholia, which is a measure of severity of depression, 85% of our sample had it. In a most similar study with looking at recurrent depression in Europe, 15% had. So we've clearly managed to get a much more severe group. And the other thing to bear in mind is that, unlike California, where most people at least know what smoking, drinking, and drugs are, the 40-year-old Chinese women we're looking at, they don't smoke, they don't drink, and they have no idea what an illegal drug is. So confounds are much reduced.
Hi, thanks for the great talk. Addressing this heterogeneity, is there a way, or based on your questionnaire, to use endophenotypes to further segregate the population?
So this is the sort of thing, it goes back to the discussion we were having a bit about schizophrenia, because people have wondered about using phenotypes for decades. There's paranoid schizophrenia, different subtypes, atypical, cycloid, psychosis, and so on. Could there be, and similar issues have gone on for many, many decades in depression. A lot of the focus about depression research is focused on reactive and non-reactive. Those people who can report their response of depression being related to a stressful life event, which is the categorization we used here. So there is evidence to suggest that distinction would work, but I think one of the things about this area rather surprised me. That when I finished collecting this sample, I thought we better just go and see what else is in the literature. There really has been nothing ever on this sort of scale. It was really quite surprising. I mean, I can understand why, because it is very labor intensive. I think maybe particularly people who only ever work in laboratories, really, which is true of me, I have to admit, until I did this, didn't really realize just how hard this is. I'll be back.
I'm quite intrigued by the cellular composition data that you showed. So given the massive leukocytosis and redistribution of immune cells and subpopulation differences in response to stress, I was wondering if you were able to or collect or have the ability to analyze before and after stress cellular composition of leukocytes.
It's a good question. We couldn't do it in our study. As I said, all we had was the DNA extracted from the saliva. So we just, there is one thing we did do, which is I thought that we could get at this by looking at the somatic rearrangements. So if we just looked at T cells, we can look at T cells and B cells, immunoglobulin rearrangements. And although our coverage is not great per person, in the entire cohort, it's pretty good. So we just count the number of recombinants that we're getting around those loci and use that as a measure of the amount of T cells and B cells. Sure. So we did that. There is a bit of a difference, not much. But I didn't show those data because it's like a little bit messy. We're not really sure what it means yet.
Cellular data, messy.
Yeah.
Right.
I'm just used to working with DNA.
Okay, Lisa.
Following up a little bit on Ming's comment about immunology, did you have any data about sort of physical comorbidity in this sample, and could you look to see whether there was a relationship between, say, the mitochondrial?
there's a number of things I regret that we didn't do, and that's one of them. We're just starting another study. My interpretation of everything I showed you is that this was underpowered. We just about saw something, and it gave us a lot of hints as to what we could do. So I had to go back to my funding agency in the Wellcome Trust and say, hey, remember that 1.5 million you gave me? Think of that as a pilot study. Would you like to? And they kindly agreed, and we took a bit of persuading, and we're now funded to collect 48,000. So so we're definitely asking about comorbidities, other disorders. We're also asking more about bipolar symptoms, hypomania, which we should have asked. We're also asking more about current state. I'm sure there's something really important we're going to forget. And if anyone can it, please tell me now, because we're just drawing up the protocol. But coming back in 4 years, and I can answer that.
So you'll be doing saliva again, or
not
blood?
Saliva. So saliva is mostly bacteria. It's fine if you just want to do genotyping, but we had to throw away about 10% of the samples because of bacterial contamination.
So you could measure, cytokines, chemokines, some immunological function in the blood samples as well as DNA, right?
Yes, exactly. And we have those options.
Do some cell sorting and get cellular composition.
Okay, thanks. One question, comment I would have about the stress data. As you know, we talked yesterday about our colleagues, George Brown and Tyrrell Harris in London, who were very, I guess, seminal in the life events field. How one disaggregates so-called independent life events from events that the actor themselves has produced. For example, being fired is a bad thing, but maybe you got fired because of your behavior, whereas certain other things like an earthquake presumably wouldn't be under your control. Do you have any thoughts about that? How to sort out those things?
Well, as you know, that's a very complicated question, and George Brown would, I know, be very unhappy about us just giving out the questionnaire and expecting us to get the
Absolutely, and you didn't rate the life events for 2 hours.
Of course not. We didn't do that.
By committee.
By committee, and that's definitely what we should do. What we decided to do, I had a long conversation with Ken about this, and we decided to try something. We don't know whether it'll work, but we've come up with what we call a life history calendar. So what we're doing this time is we're asking each of our participants to tell us how many events they've had. And we ask them then to think back to the first event and then give us an idea as to what might have been causing it. If there's nothing, then we give them a list of events we think might be important to jog their memory. And then ask them if there's anything else. We record that, we record treatment, we record response and duration. We hope that might get us a bit more of a feel for this. But I mean, we still lack the external validation that I'm sure Brown would have required.
Anyway, it's complicated, as you say. Anyway, I just wanted to thank you very much for wonderful grand rounds. We have a tradition at UCSD to present you with a little souvenir of UC San Diego School of Medicine, Department of Psychiatry. And I think in your case, it's going to be very useful because you have moved from the sheltering skies of Oxford to the searing sun of Los Angeles.
This is still in the UC system.
Yes.
I don't think they'll tell me off too much.
Wear it, and that will avoid your visit to the urodermatologist.
Okay. Thank you so much.